Hi, my name is Olivia Shirley, and this summer I worked on the Mosquito project in the Richardson Research Lab. Here on this side is one of the largest mosquitoes in the world. It's the shaggy-legged galley-nipper, also known as Saurophora ciliata. This is one of the mosquitoes that we caught this summer right here in South Carolina. So a little bit of background on testing mosquitoes here. If you've ever gotten bitten by a mosquito, you'll know that mosquitoes feed on humans, and they also include a wide variety of animals in their blood meals. The blood eaten by mosquitoes, especially human blood, may contain blood-borne viruses, which can then be tested for. A few of these viruses are herpes virus, adenovirus, hepatitis B, and HPV. Typically, a person could get diagnosed by taking a blood test, but a small sample of blood, such as what is found in a mosquito, could be tested for these diseases. So before we can monitor human blood-borne viruses, we first need human DNA in the mosquitoes. This summer was spent looking at whether we could distinguish human DNA from other mammalian DNA from various other DNA, since the first step in monitoring the community for blood-borne viruses is ensuring that our protocols work. Overall, the purpose of this project is to look for viruses in the blood eaten by mosquitoes, which can be identified through PCR analysis so that we can randomly monitor our community for these blood-borne diseases. So at this point, you may be wondering, well, what's the point of using mosquitoes? Why not just ask for volunteers? And the answer to that question is because using human test subjects requires a lot more effort for approvals and paperwork for ethically testing human subjects, and that can get pretty complicated. So using mosquitoes is a way to monitor without having to worry about those human test subjects. So that is the major advantage to using mosquitoes to monitor the community. So the research questions that we're trying to answer were, can we detect human hemoglobin in mosquitoes? Can we detect various other hemoglobin in mosquitoes? And can we detect mammalian cytochrome C in mosquitoes? So we tested these questions through a 6-step process, which was catching the mosquitoes, identifying the mosquitoes, DNA extraction, DNA purification, and then PCR and gel electrophoresis. So we caught and identified about 304 mosquitoes, and we caught them with 6 CDC light traps in 4 different locations across Horry County. And we also collected weather data and documented every insect that we caught in those traps. And the mosquitoes were then identified based on the sex, and the females were identified with the genus. So here we have quite a few examples of some insects that we caught this summer. Over here we have a glowworm beetle, which is pretty unique. It's actually a bioluminescent glowworm beetle, the adult stage. And this guy here isn't bioluminescent, but the larva is. Up here we have something that looks like a mosquito, but is actually a cranefly. This guy over here and this one in the middle are male mosquitoes. You can tell the male versus the female because the male actually have these fuzzier antennae and fuzzier, they're called palps right here. And the females are nowhere near that fuzzy, and the male actually has that to detect females around him. And then down here, of course, we have our mosquitoes right here. These are nice female mosquitoes, and this one as well. And then, of course, we have a few moths and a few other things over here. And After the mosquitoes were identified and labeled, certain mosquitoes that appeared to be engorged were given priority to be tested and were selected out of the group. You can see here the difference between two female Aedes mosquitoes. The one on the left appears to be engorged, while the one on the right doesn't. And so mosquitoes like this one on the left were given priority in testing, and so almost all of the mosquitoes that we tested this summer had some degree of engorgement, like that one on the left there. So the mosquitoes were then processed with a cycloprep genomic DNA isolation kit to extract and purify the DNA, and the DNA was then PCR tested for human hemoglobin and mammalian cytochrome C. So the first question that we tried to answer, can we detect human hemoglobin in mosquitoes? And the answer is yes. We can say that we can indeed detect human hemoglobin in mosquitoes. In this gel image, mosquito number 79 is positive for human hemoglobin with the correct size band. As you can see, the arrows are pointing to it. The band for number 79 in the first column here, and the first column in the second set of samples. Both of these bands match the expected control for human hemoglobin. So question number 2 was, can we detect various other hemoglobins in mosquitoes? And the answer to that is also yes. So as you can see in this lane here and this lane here, for mosquito number 50 for these two hemoglobin primers, we have some bands of DNA, but they don't quite match up to the control, the known control here. So the reason for that is probably because hemoglobin is a highly conserved protein. And so while it may not be human hemoglobin, it's probably a hemoglobin of something else that the mosquito fed from. And question number 3, can we detect mammalian cytochrome C in mosquitoes? And the answer to that is also yes. So in this lane right here, we have a band from mosquito number 74 that was tested for mammalian cytochrome C, and the control here, as you can see, it does line up with the control. So one of the animals that this mosquito probably fed from was a mammal. So after testing the first 36 mosquitoes and finding various results that point to the DNA being from neither human nor any other mammal, we decided to test some of these mosquitoes to see if they could possibly be from a reptile or amphibian using a reptile amphibian cytochrome C primer set that we had already had. And from this experiment, we found that 4 of the 20 samples that we tested could possibly have reptilian DNA in them. And you can see that in these wells here, that they're pretty faint bands, but you can see that they're definitely there. They're not all the same size though, which is interesting. And I will talk more about that in just a second. 4 mosquitoes that were tested matched the human hemoglobin 2 control, and 10 mosquitoes that were tested matched the human hemoglobin 3 control. So these mosquitoes, we can say, have probably fed from a human at some point. 6 mosquitoes matched the mammalian cytochrome C control, so these mosquitoes probably fed from a mammal at some point. So these first 3 columns right here in the table show the number of samples that did match our controls. And these second 3 columns here in the table show the number of samples that tested positive but did not match the controls. And a mosquito can fall into both of these categories where they both match and don't match. As you can see, mosquito number 87 here with the mammalian primer has 1 band that matches the controls and 2 bands that don't match the controls. And each color on the table here corresponds with a colored circle on the graph. So This 4 mosquitoes that tested positive for human hemoglobin 2, 2 of them are right here, number 87 and 88. So you can see that there are several examples of the mosquito testing positive for both matching the control and not matching down here. And number 779 does it right here as well. And lastly, this final column on the right is the Reptilian and Amphibian primer set that we tested on 20 mosquitoes. And so here are 10 of the mosquitoes that we tested for the Reptilian and Amphibian primer set. And as you can see, 4 of them tested positive for something. And even though we don't have a positive control for this primer set, we know that some of these bands, like this one here and this one here and this one as well, are all the same size. So we can say that it is possible that these mosquitoes, number 16, 43, and 52, actually fed off of a reptile or an amphibian. So what do these results mean? We were able to distinguish between human blood meals, mammalian, non-human blood meals, and blood meals that were neither human nor mammalian. And we also found that many of the mosquitoes that we tested had a combination of blood meals that they had fed from. So as for the future direction of the project, we plan to continue testing the mosquitoes for human hemoglobin and mammalian cytochrome C, test primers for adenovirus, herpes virus, hepatitis B, and papillomavirus, one of which is actually already in the works. Test mosquitoes that are positive for human DNA for blood-borne virus, and the ultimate goal is to monitor the community for blood-borne viruses from random blood sampling from the mosquitoes. And that is all. Thank you for watching.